अस्सलाम वालेकुम। आज का जो हमारा सब्जेक्ट है वो एनाटॉमी का है और इसमें हमारा जो पहला टॉपिक है वो सेल साइकिल के ऊपर मैं वीडियो बना रही हूँ आप लोगों के लिए। और ये सेल टॉपिक का जो है टॉपिक ये मैंने आपकी गाइटन की बुक से तैयार किया है। और गाइटन और उस एनाटॉमी की किसी भी बुक से आप इसको कंबाइन कर सकते हैं, क्योंकि फिजियोलॉजी और एनाटॉमी में ये टॉपिक जो है वो दोनों में सिमिलर ही है। तो मैंने वहाँ से मुझे ज़्यादा अच्छा मटेरियल लग रहा था तो मैंने आपके लिए वहीं से तैयार किया है। तो आप ये मेरे नोट्स बने हुए देख सकते हैं, आप इसी को तैयार कर लेंगे, आपको पूरे मार्क्स मिल जाएंगे। ठीक है। अब सबसे पहले फॉर एग्जांपल आपके पास लॉन्ग आता है लिखने के लिए सेल साइकिल के ऊपर। तो सबसे पहले आपने इसकी डेफिनेशन लिखनी है। इसकी डेफिनेशन क्या है कि द सेल अंडरगो अ सीक्वेंस ऑफ चेंजेस विच इंट्रोड्यूस अ पीरियड ऑफ ग्रोथ, रेप्लिकेशन ऑफ डीएनए एंड फॉलोड बाय अ सेल डिवीजन। कि हम क्या कह रहे हैं कि सेल साइकिल एक ऐसा इज अ साइकिल है इन विच सीरीज ऑफ चेंजेस, यानी a sequence of changes occur, and it is a whole cycle in which series of changes are occur, and which involve a period of growth, यानी इसमें हम सेल की ग्रोथ को देखते हैं। रेप्लिकेशन होती है यानी डीएनए अपने आप को रिप्लिकेट करता है, एंड फॉलोड बाय सेल डिवीजन, और उसके बाद एक सेल डिवाइड होके एंड a cell divides and it produce two into the two daughter cells। ठीक है। then two daughter cell further divided to form four daughter cells। in this way this division proceeds in a sequence of changes। ठीक है। all the cells are produced from the pre-existing cells। this line is taken by a cell theory। you have read in the previous classes that all the cells are created by the pre-existing cells। ठीक है। we cannot understand we cannot talk that cells are produced by from the non-living thing। no, it is not right। all the cells, all the living things are produced from a living thing। so cell is also a living thing, so it is also produced from the pre-existing cells। pre-existing cells undergo a cell cycle, a sequence of changes, it divide, it replicates DNA, it grow and it undergoes cell division, and then it further produce new daughter cells। ठीक है। then we discuss two phases of cell cycle। ठीक है। the two phases of cell cycles is given below, which was introduced by Howard scientists। ठीक है। Howard scientists introduced this cell cycle, and the phases of cell division are the two phases of cell division may be and three phases of cell division may be। ठीक है। the major are the two phases of cell cycle, which are interphase and M phase mean mitotic phase। ठीक है। but now in the modern study we can include cytokinesis as a third phase of cell cycle। ठीक है। so if you want to write three phases of cell cycle then there is no issue, and if you want to write two phases of cell cycle then there is no issue, but major they are the two phases of cell cycle, which are interphase and the mitotic phase। ठीक है। now we describe all these phases one by one। first of all interphase। interphase क्या है? phase of non apparent division। non apparent division mean by that in this phase there is no cell division process। the division of the cell process take place in the mitotic phase। so interphase is quite different from the M phase mean mitotic phase, because in this phase cell prepared itself for the division। ठीक है। in this phase no division occur but the cell prepared itself for the division process। ठीक है। it is a phase between the two consecutive divisions। ये it is a phase between the two consecutive division mean by that it follows by a division phase। ठीक है। it followed by a division phase, so therefore it phase between the two consecutive division। it is wrongly called as resting phase। sometimes we call interphase as a resting phase, but it is wrong, because in this phase the cell does not undergo a rest, because it is a phase in which cell become great it is a phase in which cell perform great metabolic activities, so therefore we can't call interphase as a resting phase। ठीक है। it is a period of great biological activities। in this phase cell prepared itself, cell make its proteins, its RNA, and prepare its DNA for the replication in the next phase। then Then we have a Viva question that what is a G naught phase? G naught phase is a resting phase in which cell residue until they receive appropriate signals. ठीक है, sometimes our brain send appropriate signals to a cell and allow it to undergo a resting phase, which is called G naught phase, and the cell may remain in the G naught phase for a year, for a month, and for a week. But some cells, for example nerve cells and the muscle striated cells and the eye cells, does not come out of the G naught phase. They remain total life of a human being into the G naught phase. ठीक है, and now we discuss the importance of G naught phase. That the G naught phase decrease the probability of a cell to change into cancerous cells. ठीक है, sometimes abnormal division take place in some cells and which convert the normal cells into the cancerous cells. So G naught phase play an important role in to decrease this chance of probability in the cells. ठीक है, now we discuss the types of the interface. The types of the interface are given below. First of all, G1 phase, then S phase, and then G2 phase. G1 phase can also be called as first gap phase, and it also called as first growth phase. S phase can also be called as synthetic phase, and the G2 phase can also be called as gap 2 phase or second growth phase. ठीक है, now first of all we discuss G1 phase. G1 phase, it is an important MCQ that 30 to 50% of the total time of cell cycle take place by the G1 phase. So it is the longest phase of the interface. 30 to 50 जो time होता है cell cycle का take place in G1 phase. अब G1 phase में following process take place. First of all, it is a phase of high metabolic activity. ठीक है, ड्यूरिंग दिस फेज सेल मेक इट्स एंजाइम्स. ठीक है, नंबर ऑफ क्रोमोसोम इंक्रीजेज एंड RNA फॉर्म मीन द प्रोसेस ऑफ ट्रांसक्रिप्शन टेक प्लेस इन द G1 फेज. ठीक है, डिफरेंट टाइप्स ऑफ RNA आर फॉर्म. फॉर एग्जांपल ट्रांसफर RNA, मैसेंजर RNA, राइबोसोमल RNA. all these types of ribosome RNA are formed in this phase. ठीक है, so it is a phase of high metabolic activity. We can't call that is as a resting phase. So therefore we call that interface is not a resting phase, क्योंकि इंटरफेस की फेस G1 में हमारे पास RNA is produced and the very important process, transcription process, is take place in the G1 phase. So pre-mitotic cells can also exist in this phase, can be called as G naught phase. ठीक है, G naught phase second name is pre-mitotic phase. ठीक है, so sometimes the cell undergo a pre-mitotic cell phase which is called as G naught phase. Nerve cells and the eye cell and the striated muscle cells enter into the G naught phase throughout its life. ठीक है, once if a nerve cell or eye cell or a muscle cell enter into the G naught phase, then it cannot be come out. ठीक है, then the new cells of the nerve cells are formed and then they perform their activities, and if they enter into the G naught phase, they cannot come outside and they cannot perform their function into the G naught phase. G naught phase is a resting phase. ठीक है, in this phase cell cannot be able to perform its functions. Some cells enter into the G naught phase for a year, for a month, even for the whole life. ठीक है, there different time period for the different cells to spend their times into the G naught phase. All the cells do not have a single fixed time to remain into the G naught phase. Different cells have different time. For example, nerve cells spend their whole life into the G naught phase. ठीक है, they cannot come outside. So all the cells have different time to spend some of their lives into the G naught phase, and it is also called as first growth phase, which I have explained to you. Now the next phase is S phase. It is a phase of DNA replication. In this phase, an important process takes place that DNA replicates itself into the and number of chromosome also doubles. ठीक है, in G1 phase, the chromosome is in the form of chromatin. ठीक है, then then when S phase comes, then the chromosome chromatin duplicates itself into a whole chromosome structure with the two chromatid sisters and one centromere inside itself. ठीक है, the shape of chromosome become this like in S phase, this. ठीक है, these are the two sister chromatids of the chromosome and it is the centromere of the chromosome. ठीक है, and it is also called a synthetic phase because in this phase cell synthesizes DNA, duplicates its DNA, and various type of proteins, various types of enzyme are also formed in this phase. Cell prepare itself for the division. For if S not does not place does not take place, then the cell cannot able to divide, so therefore it is very important for the cell division. Now the next is that the S phase is 35 to 45% of the total cell cycle, mean total cell cycle time का 35 से 45% take place in the S phase. ठीक है, now the other phase is G2 phase and it take place only 10 to 20% of the cell cycle. It is very short than the S phase and then the G1 phase, which are very long. ठीक है, in this phase it is a pre-mitotic phase or cell store energy, make RNA and protein. ठीक है, actually in this phase cell is ready to divide and therefore it also called as pre-mitotic phase. ठीक है, cell prepared itself for the further division. Cell prepare itself for the further division. And it also called as second growth phase or second gap phase. ठीक है, second growth phase is its modern name, but it previous its name is second gap phase. ठीक है, so now we can take its second name as the second growth phase. It is more correct. ठीक है, then we have a viva question, which is the length of each phase is different, that we cannot call that the length the time that G1 phase taken and the S phase taken and the G0 phase taken is equal. No, this is wrong. All these phases take its own time and it is not equal. G1 phase take more time, S phase take more time, but in comparison with G1 phase and S phase, G2 phase take small amount of time. ठीक है, in human average cell cycle is about 24 hours. ठीक है, for it 24 hour is the total times of cell cycle of a human cell. Or for the other cell, for example ये cell take 90 minutes, so all the cells have its fixed time to take place, so the total cell cycle time is 24 hours for a human being. Now we divide this 24 hours into the separate phases, that 30 minutes is taken by the mitotic phase while the remaining 30 minutes are taken by the three phases of interphase. ठीक है, mean that we can call as that 30 minutes is taken by interphase. And how this time of interphase is divided, 30 minutes of interphase, how divided into the G1 phase, S phase, that at follow that at 9 hours out of 30 minutes, 9 hours is taken by G1 phase, 10 hours is taken by S phase and 4.5 hours is taken by G0 phase. ठीक है, so we cannot call as 30 minutes, it's wrong. We can add up all these things and then we get the time of the interphase, which would become which become total into the 24 hours. ठीक है, okay, now I understand that this 24 hours only includes interface times, so all these when added up it become equal to 24 hours. And the time of mitotic phase is not included in the cell cycle, because cell cycle the mitotic phase is separated, because mitotic phase is a cell division process. It quite different from the cell cycle, so it quite different. ठीक है, these 30 minutes are out of 24 hours. 24 hours are only taken by the interphase, these process. ठीक है, these 24 hours are only required for the interphase, then the 30 minutes are separately allowed by the separated used by the in mitotic phase, which is a cell division process which is separated from the cell cycle. Okay, now we have a circle. Here every cell cycle of yeast is 90 minutes, which I have explained to you. Now this is G0 phase and it is time taken by the G2 phase. This is mitotic phase. This is cytokinesis. Very small amount of time is taken by the cytokinesis because it is only division of the cytoplasm. So there is need of small amount of time. Very little time is required for the cell cytoplasm to divide into the two daughter cells. Then G1 phase required a lot of time and the S phase required small amount of time for the DNA synthesis. Okay, this is the S phase. Another graph shown, this is S phase, a whole big phase. This is G1 phase, this is M phase, and this is G2 phase. G2 phase is quite short than the G1 phase. G1 phase is more than the G2 phase. Okay, so do not consider them equal time. These are quite different times. Okay, X phase is quite long, so I explained this that X phase is also long than the G1 phase. Okay, in this diagram. Now we further proceed. Yes, now I have explained some things about the microfilament. It is quite separated from the cell cycle topic, so take it take this concept. फर्स्ट ऑफ ऑल माइक्रोफिलामेंट्स किस में अपना प्ले अ इम्पोर्टेंट रोल इन द सेल डिवीजन. ठीक है, प्रोवाइड स्पेस टू द ऑर्गेनेलीज, साइटोकाइनेसिस and diappendicis. Yeah, means diappendicis mean by that the white blood cells move out of the blood vessels from the limb. We say that sometimes our from the interstitial spaces, the white blood cells and the various different kinds of protein move out of the blood. Okay, and then they become the lymph. Okay, and lymph is quite different from the blood because lymph does not contain red blood cells. Okay, so it is different from the our blood. Okay, and this lymph become a part of our lymphatic system. So this process is called diappendicis. Okay, and microfilaments play important role in this diappendicis. Okay, then microfilaments have two types of important protein. First of all is tubulin and the other one is actin and myosin. But they important play role in the cycle division. They maintain cytoskeleton cell structure. Okay, they are tube like structure which are present in our cell and they maintain our cell shape. Okay, they important maintain the cell structure. And actin and myosin play important role in phagocytosis. ठीक है, we study the process of phagocytosis and we see that the cell have actin and myosin around its membrane inner side. ठीक है, so actin and myosin allow the cell membrane to become dilated and to enter the food particle into it, and this process take place due to actin and myosin protein which are present inside the cover of cell membrane of the animal cell. Now we have intermediate filament. What the important play role, that cell inheritance to cell or extracellular matrix of ECM यानी एक्स्ट्रा सेलुलर मैट्रिक्स. सो क्या होता है कि हम various cells are required to transfer their material from one cell to another cell, so they have required that one cell can be attached to the other cells for the easy transportation. So intermediate filaments play important role in this attachment to cell, so therefore easy transportation can take place, and it also help in the cellular movements, means cytolysis. ठीक है, mean that in cells all the organelles move around about the nucleus. So in this movement intermediate filaments play an important role in this movement in cytolysis. ठीक है, now next we have general events that take place in the interphase. And what are these events? If you make a separate column and make it in different kinds of color, then you will easily memorize it. First of all, the event that occur in the interphase is that a nuclear envelope remain intact, mean nuclear envelope remains same. That we see in the process of cell division, nuclear envelope break up itself, and then the whole process of cell division take place. But in the process of interphase, there is no cell division. It is quite different from the mitotic phase. So therefore, the nuclear membrane remains same, remain intact in this process. Now the next thing is chromosome are in the form of chromatin material. ठीक है. In interphase, chromosome is remain same as a thread like structure which is called chromatin.